AQA Biology. Key terms. DNA sequences involves reading base sequencing of a length. DNA hybridization, technique to determine similarities between DNA sequences and different organisms. Biodiversity, variation of species. Biodiversity hotspot, lots of different species in an area. Endemism, an ecological state of species being unique and in a defined geographical location. Techniques refining our ideas and classification. DNA sequencing, DNA hybridization, and amino acid sequencing. So then, basically, DNA sequencing is also known as chain termination sequencing, where you have a DNA sample, primer, 4 nucleotides, and DNA polymerase. You add in each test tube, you add a radioactive nucleotide, and then let the DNA polymerase synthesize the nucleotides, creating small fragments which stop at the little radioactive nucleotide, which is represented by a little star, which would look like this. I know there's a word for this, but I can't pronounce it, so I'm not even going to attempt. And the sequence can be read off starting with the smallest fragments. So you'll have a little diagram that will look like this, with your little A, T, C, G, and different lines representing the different fragments. So you might have one here, one here, one here, one here, and then one here. So this will be read A, G, C, T, A, because the shorter the fragment, the further down the plate it goes. And then you have DNA hybridization. DNA hybridization is when DNA separates when heated and then recombines to form the original strand and then is left to cool. All strands within the DNA mixture will pair up with their partners eventually. And then because they pair up with the things that they are complementary, if you add a radioactive matter to the system, then it means that when they recombine, they may recombine with a radioactive substance, which will form a hybrid strand, which is 50% radioactive and 50% labeled DNA. So then what you would do, at each temperature, you would heat it, and depending on the temperature in which it can withstand, it will depend on the amount of hydrogen bonds which form between the radioactive and the DNA strand. And the uses of hybridization is used to classify like flowering plants and such. And amino acid sequencing is when amino acid sequences are determined by DNA, and similarities between them represent similar proteins and how they're related. The disadvantage is that as the amino acid code is degenerate, there could be more than one sequence for this. So what you would do, you would get, so as things such as antibodies and proteins are all made up of amino acids, you would add, so what you would do this via the use of injecting antibodies. So what you would do, you get the serum and you would inject it into an animal. And this animal would produce antibiotics to fight off that infection in which you wanted to fight off. Then the serum would be extracted from that species and mixed with blood from the species in which you're going to inject it into. Then once you inject it into that animal or person, if the body doesn't fight against it, that means that our DNA and antigens are of a similar type of shape, because your body automatically wants to fight anything foreign. So if you have similar sequencing, that means your body's less likely to want to fight it. Biodiversity. The importance of biodiversity is that it's the diversity of the living organisms. And the components of biodiversity are species diversity, which are the number, abundance, and most number and abundance, and more species give a higher diversity. Genetic diversity is a diversity of different aspects and variations of alleles, and ecosystem diversity, which is the variety of different habitats. More different habitats equal a higher level of diversity. Now that's all there is for this topic right now, and the next video will be about quantitative investigations of variation, which will be about sampling methods, how to measure species diversity, and threats to species diversity. So if you like this video, then please like and subscribe, and I'll see you on the next one.